tolerance for promiscuity in terms of gene exchange tends to be higher the less obligate the sexual exchange, and also the less often the organism has to participate in sex in order to, for example, reproduce. Thus, bacteria tend to have a much higher tolerance for promiscuity in terms of what kinds of DNA they can acquire versus the ability of oppositely sexual organisms, particularly in terms of the mating that gives rise to their reproduction, are capable to tolerate promiscuity. We can also consider the degree to which tolerance for promiscuity can be associated with the potential for genes that are coadapted to be physically linked together. In those organisms that display a low tolerance for promiscuity, it's possible for genes to be coadapted even if those genes are not physically linked together. On the other hand, for organisms in which a high degree of promiscuity is observed, in order for genes to move from one organism to another organism, if those genes in fact are coadapted, it's unlikely that in the recipient organism the necessary associated alleles will already be present, and therefore those genes must be moved as one block from organism to organism in order for them to be functional in a recipient organism and therefore have the potential to survive in the face of natural selection. It's only when genes are physically linked together in blocks that their likelihood of being transferred via horizontal gene transfer from one organism to another is high. Therefore, in those organisms that display a relatively high amount of promiscuity, you also see a relatively high amount of linkage of those genes that are coadapted, and this is because those genes that are not linked together that nevertheless are coadapted are less able, perhaps much less able, to be distributed around communities as a consequence of horizontal gene transfer, and therefore have the potential to be less represented within environments, within communities, than those genes that in fact are physically linked together.